Hello, my name is Paul Goepfert. I am a professor at the University of Alabama at Birmingham. I'm in the Division of Infectious Diseases. I've been involved in clinical vaccine trials since 1997, and in the past year, I've been heavily involved in COVID-19 vaccines, and I'm a co-chair of the J&J vaccine efficacy study known as Ensemble, and I'll talk about that in a little bit. And I'm very excited to be able to present to you today. So I'm going to start with a slide that talks about the vaccine candidates in development for SARS-CoV-2. And obviously this has been wonderfully successful now. And what I'm highlighting here, and you see this in green, is the ones that have been shown to work in one way, shape, or form. And you can see that with the exception of one. So Pfizer and Moderna are the RNA vaccines. Novavax and Sanofi are the protein-based vaccines. The Sanofi, I won't talk about too much, but they had to go back and redo their vaccine because it wasn't purified enough, and they're now just getting back started on that one yet. AstraZeneca, Janssen, or J&J, and Sputnik V vaccine all use this recombinant adeno-type platform that I'll talk about. And Sinovac uses an inactivated viruses, which is one of our oldest technologies. But most of these vaccines, with the exception of Sanofi, have all been shown to work in varying degrees, and I'll talk about that later. So just to back up, one thing that we've known for a long time now is that antibodies are very important in protection for vaccines. So the gold standard is actually what are called neutralizing antibodies, but you can also induce functional antibodies to a certain extent as well. However, most people in the field think that neutralizing antibodies are what you want for a vaccine to induce in the person getting vaccinated in order to prevent infection. And so what are and I'll just say that these, the polio, measles, rabies, diphtheria, tetanus, neutralizing antibodies are by far and away the most important correlate of protection. But then all of these on here have been shown to be correlates of protection in terms of just antibodies alone. So that's what we really wanted to get with these vaccines to begin with, ideally. So neutralizing antibodies, what are they? So this is a diagrammatic representation of that. So you have SARS-CoV-2 that wants to go into the cell, and what it does is it attaches to the ACE2 receptor. The ACE2 receptor, just parenthetically, is on many, many cell types. And so that's one of the reasons they believe that SARS-CoV-2 and COVID-19 is such a devastating disease for some people, because it can attach to many different cell types. So anyway, the virus does that. It gets into the cell, and then it does what viruses do, which is make more viruses inside that cell. What neutralizing antibodies do is they actually will bind to the virus and prevent it from attaching to its cell receptor, the ACE2. When you can prevent it from doing that, viruses don't last long unless they go from cell to cell, and so they don't last long and they end up dying off. And so that's why you can actually neutralize the virus with these neutralizing antibodies. And so you can think of neutralizing antibodies as sort of a spear that it can kill the virus upon attachment. The other kinds of antibodies, which are just binding antibodies or functional antibodies, you can look at those as being a net that they can sort of keep the virus at bay, but they can't kill it, and they need extra help to kill that virus. So I'm going to talk a little bit about how mRNA vaccines work, because these are the first two vaccines, Pfizer and Moderna, that have been shown to be effective in the United States. And so, interestingly enough, SARS-CoV-2 encodes 30 plus different proteins, but we knew from even before COVID-19 that the spike protein is the Achilles heel of coronaviruses. So obviously SARS-CoV-2 is a type of coronavirus. We have seasonal coronaviruses, but we knew when SARS-1 came about in around 2003 that many studies were done showing that if you can target the spike protein, that the virus, it sort of cripples the virus. So we knew that very early on, and so the companies, Moderna and Pfizer, knew to use this protein. Another group, the NIH, a guy by the name of Barney Graham, figured out that if you can actually do a certain form of the protein, the spike protein, it's even more immunogenic. It induces even better immune responses. And so Pfizer, Moderna also used that specific protein that was highly immunogenic. And what they did was they then very quickly got the spike genome, the mRNA that only encodes for the spike protein. So then what happens is they just take that mRNA that encodes the spike protein and they encase it in a lipid nanoparticle. And the reason they have to do that is because the mRNA is extremely labile. And it probably is naturally labile because mRNA, as you probably know, is used by cells as a genetic template to make proteins. And it doesn't need it after it makes the protein. And so cells just get rid of it very quickly afterwards, and so does the environment. So in order to make this stable, they have to put it in a lipid nanoparticle. And as you know, for mRNAs, one of the biggest problems with these vaccines is they require very cold storage, even with the lipid nanoparticle. So nonetheless, you have this lipid nanoparticle that encases the mRNA, and you inject the mRNA lipid nanoparticle into the arm of a participant. And then in that arm, in the muscle, there are mainly muscle cells there. So this particle is taken up by the muscle cells, and then muscle cells do what they normally do, which is make proteins when they're given a genetic code to make that protein. And it'll make proteins, and it'll make, in this case, the SARS-CoV-2 spike protein. And so you're using your own cells as a protein production factory, which is actually a very ingenious thing to do because it can be difficult to make proteins in the lab. Instead, in this case, you're using everybody's own cells to make proteins. So now what happens is these proteins then are taken up by what are called antigen presenting cells, or APC. And these APCs then go to lymph node and they interact with T cells and they interact with B cells. And the T cells actually can help the B cells make really good antibodies. And then these B cells make antibodies, in this case, that hopefully will neutralize SARS-CoV-2. What's interesting is these mRNA vaccines can also make T cells that can actually kill virally infected cells. And so even if the neutralizing antibody doesn't necessarily get rid of all sort of virus before infection, even if the person gets infected, you can have T cells that can sort of mop up that infection. And so this is ideally what you'd like for an mRNA vaccine. I'm also going to talk about recombinant adenovirus vaccines, and this is the one from J&J, and also AstraZeneca has a vaccine that is also based on similar technology. So in this case, what you use is you used an adenovirus that is made replication incompetent. So it's only used as a Trojan horse. And inside that Trojan horse, in this case, you have DNA. So the adenovirus gets into the same muscle cell and it uncoats, and then you have the spike DNA. And again, cells in this case have to go from DNA to RNA, and then the RNA makes proteins. And then the same thing happens. The protein then gets taken up by antigen-presenting cells, which go to the lymph node. And in the lymph node, the antigen-presenting cell interacts with T cells, both CD4 helper cells and B cells and killer T cells. And you get the production of hopefully neutralizing antibodies and these killer T cells that can mop up infection. So in essence, the mRNA vaccines and these recombinant adenovirus vaccines really do work in the same manner. They just do it mechanistically a little differently. So why is it that these vaccines have been developed so rapidly? And the short story is that they have all undergone rigorous safety testing, just like most any other vaccine has undergone rigorous safety testing. However, what's different about these vaccines is that we've been able to rapidly develop them because of certain factors, and I'll show those here. So first of all, this is the timeline. I'm using the Moderna vaccine as an example. So December 31st, the Chinese reported COVID for the first time, and 10 days after that, they actually made the sequence of SARS-CoV-2, which is the virus that causes COVID. They made it publicly available. And companies, Moderna, at the time, it took them 7 days then to actually make a vaccine, which is phenomenal. If you use old technology, protein-based vaccines, it will take you several months to years to make a vaccine, once you know the actual sequence. So they made it very, very quickly. And they tested it in small animal models and found that it was immunogenic and it seemed safe in those small animal models. And so then just a few months later, they did their first phase 1-2 study, first in human study, where they tested safety and immunogenicity. And then what's really important as well is they got government support. So the US government put a lot of money into the development of Moderna, AstraZeneca, J&J, Novavax, and Sanofi vaccines. And this allowed them to rapidly start efficacy studies. And these were started at the end of July and middle of July for Pfizer, as a matter of fact. And so then they were able to use that, and they were able to get some sites that for free for them. They didn't have to pay for some of the clinical studies because, again, it was government supported. The government gave more money. They promised more money to help them develop the vaccine if it became an effective vaccine. And then, as you know, in the middle of November, this vaccine was shown to be 94% effective, and I'll show you that, in preventing COVID. And then they got approval a month thereafter, and that's when the vaccine started rolling out. So why was it developed so fast? Just to reiterate this, we already knew that targeting the spike protein was extremely important. We also rapidly identified and sequenced the virus. RNA vaccines can be rapidly developed, and so we were very lucky that that technology was available and ready to use. There was some very smart thinking in combining both phases 1 and 2, which had helped move things along much, much more quickly. And the other thing is that we rapidly enrolled participants. And this was a huge boost from the community. So the community realized how important it was to get vaccines. And we had huge numbers of volunteers that were ready to go for these studies. And so we enrolled at least 30,000 people in each one of these studies, and up to 45,000 for the Pfizer and J&J studies. And then sadly, the other thing is that we continue to have huge numbers of new COVID infections. And you need new infections in order to show that a vaccine worked. And because we had so many, it would allow us to get our answer much more quickly than we normally would. So those are the reasons, and I'll point out that none of these say that because we didn't test them for safety. And I'll note that the 30,000 individuals that you test for an efficacy is as much, if not more, than most vaccines have actually tested to become FDA approved. So I'm focusing on now the mRNA vaccine from Moderna, and this was the phase 1-2 study. And what this is then is a demonstration of neutralizing antibodies, a graphic demonstration of that. And so on the y-axis, you have the titer of neutralizing antibody, and obviously the higher up it is, the better the neutralizing antibody. And on the x-axis, you have the different doses of vaccine, so 25 micrograms to 250 micrograms. And then this vaccine was given at day 1 and day 29. And so what you see then, I'm going to focus on the 100 microgram, the one in the middle, is that after the first dose, you do get some amount of neutralizing antibody. But if you compare it to the convalescent on the right, it's less than the convalescent. The convalescent are people who've recovered from COVID infection. But once you give that one boost, the neutralizing antibodies, as you can see at days 36, 43, and 57, are actually higher than people who've recovered from COVID. And so you get better antibodies with the vaccine than you do with natural infection. And this has been shown for Pfizer as well, and several other vaccines. And so that's one of the reasons we recommend that even if people have been infected previously with COVID, they should still get vaccinated. So at any rate, what happened was that the 100 microgram dose was the one that was well tolerated and gave as good of an immune response as the higher dose and better than the lower dose. So this was taken forward to an efficacy study. So how do we do efficacy studies? We vaccinate 30,000 people, half of which, 15,000, get the mRNA vaccine, and the other half get placebo. And importantly, it's a randomized double-blind study, which means that neither the person giving the vaccine nor the person receiving the vaccine nor the investigator knows what they get. So they don't know if they got the vaccine or if they got placebo. So they get a couple of injections, 28 days apart, and then what happens is you have to rely on natural exposure to see what happens. And at the end of the period of evaluation, you hope that the people who got the vaccine have less COVID than the people who got placebo. So I'll talk next about the local adverse events in this vaccine. And what you can see is that the majority of people had local adverse events. So any adverse events, just focus the one on the left. That over 75% to 80% actually had local pain, and then some had erythema, swelling, and lymphadenopathy after the first and second doses. However, these were transient. They were generally mild or grade 1, and they went away in 1 or 2 days. So then they also looked at systemic events, and what you can see is the systemic events are a little bit more significant in that you can see that after 2 doses, again, the majority of people had some sort of systemic event. So these things include fever, headache, fatigue, things like that. The most common ones were headache and fatigue. And you can see that most were actually mild to moderate, and there were some severe. But again, the vast majority of these go away in 1 or 2 days. And then the participant had no other further problems. So next is the efficacy slide. And the way you do these then again is you follow them over time to see who gets symptomatic COVID, PCR positive detected COVID. And in the gray line is the placebo group. And you can see that on the y-axis, that's the cumulative event of symptomatic COVID infection. And on the x-axis, it's days since randomization. And what you see is that the placebo group has much, much higher numbers of COVID infection than the mRNA group shown in blue. And you see the arrows show when the persons were vaccinated. And what's interesting is you can see the line starts separating before the second vaccine. And so, in fact, it looks like around 12 to 14 days it starts separating. And you can make that calculation as well. And the vaccine is 90+% effective after just the first dose, and 95% effective after 2 doses. And real world data is bearing this out as well. So beyond this efficacy study, both Moderna and Pfizer vaccines have shown that after just 1 dose, it's at least 80% effective, and then 90+% effective after 2 doses. So the next vaccine that I'll talk about then is the J&J vaccine, this Ad26-CoV2.S vaccine. It also induces neutralizing antibodies. And this is a busy slide. This is their phase 1-2 study. Again, first in human study where you test safety and immunogenicity. And what I want to focus on is the low dose placebo, because that's what went on to efficacy studies. So you can see that with the low dose placebo, so all they get is 1 low dose of vaccine, and you get neutralizing antibodies that are shown there in the sort of the second group of individuals in the green color, the light green color. And what you see is that you get a certain amount of neutralizing antibodies, but compared to HCS, which is healthy controls who have recovered from COVID, that you get about the same amount of antibodies, but not better like we saw with the Moderna vaccine. Interestingly, if you give 2 doses, you actually do get higher antibody responses. But the company made a decision that because of we were in a pandemic, that they wanted to be able to just use the single dose to rapidly show whether or not this works and protects against COVID. And so they did the same study, and I won't go belabor this too much, but in this case, they enrolled almost 22,000 per arm. Just after 1 injection, they looked at 14 to 28 days, and then they looked for efficacy. So the first thing I'll show you then is the local events, and I want to point out here that the local adverse events were actually lower than with the Moderna and even the Pfizer vaccine that I'm not showing. And you can see that younger people tend to have higher local events than older people, but yet they were below 60% in this case for local events. And for systemic events, you can see that it's less than 60% as well. And you can see that the top is the actual vaccine, the bottom is the placebo. And when you subtract placebo, you can see that the local events are actually fairly reasonable. If you're talking any adverse event in an older individual, you can see that it's 45%, but the placebo is about 35%. So only about 10% people had any kind of systemic event. So the bottom line is this vaccine is better tolerated than the mRNA vaccines. So then now we're looking at the efficacy, and this is the same kind of data that I showed before. And this is any COVID infection. So this is vaccine efficacy against any type of COVID infection. And in this case, the blue line is the placebo, the red line is the vaccine. And what you can see then is that in this case, again, after 14 days, you start seeing separation of the lines, meaning that that's when you start seeing efficacy. And the efficacy continues to increase. And in this situation, it was about 70% effective. So a bit less than the Pfizer and Moderna, but that they didn't compare the two, so it's difficult to know if it's truly less efficacious. And then more importantly is vaccine efficacy against severe disease. And again, you see this, what I want to point out is that now you see separation after 7 days on the left. And the vaccine efficacy against severe disease was 80+% after 14 days. But then what you can see then is that the vaccine efficacy continues to increase in the graph on the right. And 70 days afterwards, against severe COVID infection, it was nearly 100% effective. And what's not shown here is that the J&J vaccine and actually most all the other vaccines are about 100% effective against preventing severe COVID infection that requires hospitalization or leads to death. So extremely effective vaccines. So the other thing about the J&J vaccine that was remarkable was it tested against these emerging variant strains, including, and you can see that the vaccine efficacy against severe and critical disease, as I mentioned, was 80+%. And you can see that in the United States, 86%, 88% in Brazil, and 82% in South Africa. And South Africa is a situation where they have this B1351 lineage that actually is partly vaccine resistant. But despite the fact that it's partly vaccine resistant, it was still 82% effective in preventing severe critical COVID and 100% effective against hospitalization and death. So this was the first time showing that even though we have these potentially problematic viral variants, it still worked well against them. So the other thing that a lot of people ask is, well, you're preventing against symptomatic COVID, but are you preventing against all infections? And the reason that's important is you want to be able to prevent against subsequent viral spread in people who don't even know they have infection. And this is some of the first data, but other companies have this now, but J&J was the first to really have this. And that is that you show that, that in this situation, the J&J vaccine prevents actually asymptomatic or undetected COVID-19. And what you can see if you look at the bottom panel that it actually prevents seroconversion of SARS-CoV-2 in patients and participants without any previous symptoms, and it does so in an efficacy of 75%. So not only are these vaccines preventing against COVID-19 infection where you're symptomatic, but it actually prevents SARS-CoV-2 infection outright, which is great news for the world. So this is just a slide showing what we have now in terms of the effective studies showing efficacy to date. Some of these have been published, some of these have just been given in press releases, but I show them here. You see that the Pfizer-Moderna vaccines, its efficacy rates have been 94 to 95% effective, and real-world data now, because these were the first to come out, have shown that this is continuing to be this good. I will mention parenthetically that Pfizer just this week reported that their vaccine in 12 to 15-year-olds in a small study was 100% effective against preventing symptomatic COVID. And they also did another small study in South Africa with these variant strains. And again, it was 100% effective, remarkably, in preventing infection against these variant strains. So that's fantastic news. AstraZeneca and Janssen and Sputnik V are all these recombinant adenovirus vaccines. And you can see that their efficacy rate varies anywhere from 60 to 90%. And I've talked about Janssen's vaccine and how it prevents against severe disease. And then this Sinovac and Covaxin, which are two vaccines, one made by China, one made by India, that are using really old technology to use this inactivated SARS-CoV-2. And you can see that its vaccine efficacy varies anywhere from 50 to 78%. And there's no data that I'm aware of preventing against a severe disease, but I suspect that it will as well. So I'm going to end to try and answer this question, which is, do I need to be vaccinated every year? And that is the big question. And the answer is unknown at the moment. And it's going to depend on the amount of viral variants that are circulating in the population and the duration of immunity. This slide shows back to that old schematic SARS-CoV-2 representation, and it shows a representation of the viral variants. What they've done is they've made these viral variants to test them in the test tube, and in the test tube there is a slight decrease, if you see, on the wild type versus this viral variant, which is the one in South Africa, which is somewhat vaccine resistant. However, what you see is it's just a slight drop off, and you still do get neutralization to a certain extent. And I will say that we now have vaccine data from 3 different companies: Pfizer, Novavax, and J&J, and they show varying vaccine efficacy. And AstraZeneca as well, varying vaccine efficacy against these variants. The AstraZeneca vaccine did very poorly against this variant, about 10%. The Novavax was 50% effective against this. The J&J vaccine was 65% effective against symptomatic COVID and 85% against severe COVID. And then, as I mentioned, the Pfizer vaccine was 100% effective against symptomatic COVID infections. So we'll just have to see if these variants take hold in the world. If other variants that are more vaccine resistant take hold, we'll just have to see. And then the other factor is, well, how long do these antibodies last that you give with the vaccine? And this data was published in the New England Journal from Moderna. And what you can see is, especially in young people, these antibodies continue to last at least 4 months after vaccination. They seem to decline maybe a little more in older people, but they're still present. I will say that Pfizer, which has a similar mRNA vaccine, has efficacy data now 6 months later and still highly effective at 6 months, which is, again, fantastic news. And so just to include, I think the important thing is, how is it that COVID vaccines have been rapidly developed, beating the prior vaccine record by 4 years? So the mumps vaccine was developed in 5 years, which was the prior record. And again, we had prior SARS-1 knowledge that helped us know that the spike protein was the Achilles heel of the virus and to target that. We had new technology, and remember, the mRNA vaccine came out and was approved before we even started a protein-based vaccine study. So this new technology helped us as well. New trial design, rapid enrollment into very large trials, and then the growing pandemic was helpful. And then when the vaccines were studied, we had a relatively low viral mutation rate. And it's important to say that even though these vaccines have been shown to be very effective and safe in these early studies, we're continuing to look when we start vaccinating millions of people for potential vaccine side effects and to be sure that these vaccines are still working really well. And with that, that's all I have, and I really appreciate your time. Thank you.